In today's lecture, we're going to discuss proteins, especially the proteins' compositions, their structures, as well as their functions. In chemistry, an amino acid is any molecule that contains both an amine, which is NHR2, where the R is a functional group. It also can be NH2R or even NR3 too, and a carboxyl functional group. But in biochemistry, this term amino acid refers to specifically alpha amino acids, where the alpha amino acids are the molecules where the amine or the amino group as well as carboxyl group are attached to the same carbon, which is called as alpha carbon. And it also have a side chain, which is indicated here as an R, that has some variations too. It can be just simply the hydrogen atom in glycine, or it can be a large heterocyclic group in tryptophan too, and also phenyl group, or a long hydrocarbon chain too. And it changes the property of these amino acids. And in the protein chain, where there are several amino acids, or even hundreds or thousands of amino acids are linked to each other, each of these amino acids are called amino acid residue. Two amino acids can be linked, especially through a carboxyl group from one amino acid and an amino group from the other amino acids by the bonding mechanism called the peptide bond. And when they are connected, it looks like this with the two amino acids linking together, and it's called the polypeptide. And also when the polypeptides are get together to make the long sequence of the amino acids, then it'll be called as a protein. Here's the basic structure of amino acid, which shows the tetrahedral arrangement of all the side chains that are attached to the central carbon atom, which is called the alpha carbon. And this alpha carbon is chiral, and that means this molecule can generate a mirror image too, which is not superimposable to each other. But there's an exception where this R, the side chain, is hydrogen atom in glycine. And in that case, here's H and here's H2, so it's not going to be any other mirror images. And all amino acids have COO-, which is carboxyl group, as well as NH3+, the amino group, at physiological pH, which is around 7.2, where the both functional groups are ionized and they are in the spiralion form. And this spiralion is basically a molecule containing both functional groups with opposite charges within the same molecule. And compounds like these are also called amphoteric, which means that they are both basic and acidic too. And amino acids are never neutral in aqueous solution because of this spiralion forms or the amphoteric property. And in previous lectures, we already discussed about pKa, or the pK, which is acid dissociation function. And in amino acids, they have at least two pKa's, which is called as pK1 and pK2. And also, you remember the sigmoidal curve of the titration curves. And the pK is basically right in the midpoint of that titration curves. And also, for the multiple curves, you can get multiple pK points. In general, pK1 is around 2.2 for amino acid, which is pretty acidic. And the second pK, which is pK2, is around 9.4. But these values are somewhat different from different amino acids based on the side chains that they're having or the conditions that they're in. And both pK1 and pK2 are affected by the neighboring groups. So, for example, the carboxyl group of glycine, which is the smallest amino acid, is more acidic than expected because that carboxyl group is stabilized by the amino group that is close by the other glycine molecule. And the titration curves gives two defined legs, as you can see here, as it has both two pKa's. and it's adequately described by the Henderson-Hasselbalch equation, which shows the relationship between pKa and the pH. The pH, when the amino acid has no net charge, is called Pi, which stands for isoelectric point. And for amino acids with no ionizable groups on the side chains, such as glycine, the Pi is the sum of two pK values divided by the 2, so you get the average of the pK values. But for the basic amino acids, such as lysine, arginine, and histidine, which have basic groups on their side chains, the Pi is now pK side chain, which is more basic, plus pK2, the most basic pK value, divided by 2. On the other hand, for acidic amino acids, such as aspartic acid or the glutamic acid, which have another carboxyl group on the side chain too, the Pi value is now the pK1, more acidic pK, plus pK side chain, another acidic pK value, divided by 2. And these Pi values can be derived for the whole protein too, where you take into account all the amino acids within the protein. And it is important to know the Pi value, especially for the protein purification from the really complex extracts where there are all different species of or the different types of the proteins exist. And protein has a minimal solubility when the pH value is the same as the Pi value, so you can use this fact to precipitate a specific protein in interest. And there's a protein separation technique called isoelectric focusing, which uses a difference in Pi values among different proteins as basis for separation. There are 20 standard amino acids found in the proteins in the cell, and they can be grouped based on the nature of the side chains. So 9 amino acids are nonpolar, 6 are polar uncharged, 3 are basic, and 2 amino acids are acidic. And you must memorize all the structure, the single letter and 3-letter codes of the amino acids, and you can refer to table 4-1 in the textbook. Here are some examples of the amino acids with their side chains indicated as a structure. The amino acids with the nonpolar side chains are alanine, which has a methyl group as a side chain, isoleucine, which has a hydrocarbon, or the phenylalanine, which has a phenyl ring. And also, amino acids with the uncharged polar side chains include serine and glutamine. And amino acids with charged polar side chains include aspartate or aspartic acid and lysine too. In majority of globular proteins, which are the soluble proteins, the nonpolar amino acids locate inside of the protein due to the hydrophobic effect, so they can avoid the contact with water. And the polar amino acids are located outside of the protein because they are hydrophilic. And there are some other groupings for amino acids too. Here are 2 examples. The first one is aromatic. And aromatic meaning that has the phenyl ring within the side chain, which include tryptophan, phenylalanine, and tyrosine. And also, the amino acids sometimes have a sulfur containing, which are cysteine and methionine. And cysteine is particularly important and interesting because it makes the disulfide bridges, which is critical for protein folding that we're going to discuss later on. And there are 1-letter codes for amino acids too, and they were developed and designated in a way that people can easily remember. Typically use the first letter of the name of the amino acids, such as glycine, and it's indicated as G. But sometimes the second one too, as in tyrosine or arginine. And sometimes the mnemonic rules are used too. The examples include aspartic acid, glutamine, phenylalanine, asparagine too. And these are the amino acids with aliphatic side chains, meaning that they contain alkane groups. Glycine has the hydrogen atom, alanine has the methyl group, valine has the isopropyl group, Leucine and isoleucine have longer hydrocarbon chains. And as we move from the glycine to isoleucine, the side chains in the R groups become more extended, as you can see, and as you can expect, more hydrophobic too, which is measured by the tendency to transfer from water to a hydrocarbon solvent. And more hydrophobic amino acids are usually buried within the protein molecule, so you can avoid the contact with water. And proline is an interesting amino acid because it has a cyclic side chain, as you can see here. And proline is sometimes, but rarely, also called as an amino acid because it has an amino group. And the R group is aliphatic, but it's less flexible in a peptide chain, as you can imagine, because it's linked to both alpha carbon as well as amino group 2. And proline residues are not easily folded into protein structures, so its property is actually used in some other folding mechanisms for protein structures, which we're going to discuss later on. And these are the amino acids with the sulfur or hydroxyl group containing R groups. Serine has a hydroxyl group, cysteine has a sulfide group, threonine has the hydroxyl group in the middle of side chain, and methionine has the sulfur atom in the middle of this hydrocarbon chain. But none of these R groups are ionized in the pH 6 to 8 range, which is a physiological range. But in the peptide, the cysteine group will ionize at pH around 8.5 to 9.0, a little bit basic condition. Therefore, they are uncharged under physiological conditions. Again, they're not ionized, so they're not charged. But the R group is weakly polar, so these amino acids are generally more hydrophilic than aliphatic amino acids, such as alanine of a comparable size, except for the methionine, which is still quite hydrophobic. And cysteine has the special property of contributing to disulfide bonds with the other cysteine amino acids within and or between polypeptides that we are going to discuss much more in later of this lecture. And these are the aromatic amino acids, phenylalanine, tyrosine, and tryptophan. And especially the tryptophan ring can stack with each other inside the proteins and can interact via van der Waals interactions to stabilize the internal structure of the protein. And phenylalanine is one of the most hydrophobic amino acids, as you can imagine, that has the phenyl ring here. And these two amino acids are also hydrophobic, but their hydrophobicity is somewhat moderated by their hydrophilic R groups, such as hydroxyl group here too. And these are the acidic amino acids, they're amides, as well as basic amino acids. All of them are hydrophilic, meaning that they're polar, and they're usually found on the surface of the protein, so they're exposed to aqueous environments. And the acidic and basic amino acids are charged at or near neutral pH, which is a physiological condition around the pH 7 or so, and therefore they contribute to the overall charge of the proteins. So aspartic acid and glutamic acids are the acidic amino acids, and asparagine and glutamines are the amides of the acidic amino acids. They're basically uncharged, but they are hydrophilic due to their polar side chains. And these are the basic amino acids, the histidine, glycine, and arginine. And here's a quick summary. Hydrophobic amino acids are insoluble in water, so they tend to cluster on inside of the proteins. And these hydrophobic amino acids include the ones that have nonpolar or hydrocarbons as their side chains, such as alanine, valine, and so on, and also the amino acids with the aromatic side chains, such as phenylalanine and tryptophan too. On the contrary, the hydrophilic amino acids have ionized or polar side chains, and ionized side chains are going to happen at a neutral pH, and the polar side chains include the amides or the hydroxyl groups. And they can participate in the hydrogen bonding, meaning that these amino acids, especially the side chains, can interact with the other polar groups or the water molecules itself. So the examples of hydrophilic amino acids are here, including basic and acidic amino acids, as well as polar and uncharged amino acids. And they are usually bound at the surface of the soluble proteins, and if they are located within the interior of the folded proteins, they are usually hydrogen bonded to the polypeptide backbone or some other polar amino acids. And these are special amino acids. The cysteine that makes the disulfide bond with the other cysteine, and glycine, the smallest amino acid that does not have chirality, and proline that has a rigid structure. And let's discuss more about these special amino acids. The first one is cysteine, can be written as cis or C, has this special sulfhydryl side chain, which actually enables to interact with the other sulfhydryl group on the cysteine on the other amino acids, and they can form the disulfide bond like this SS bond, which is pretty strong bonding, and they often plays important role in protein tertiary structures to link the two or more of the polypeptides within a protein. The next one is glycine, written as gly or G, has the hydrogen atom as the side chain. And this glycine is the only amino acid having a symmetric alpha carbon, so it does not have chirality again. And glycine is the smallest and thus the most versatile amino acids. So glycine can be readily fit into any protein structural elements because it has the least steric hindrance. And it's often also excluded from the alpha helices, which is the basic structure of the polypeptide. And this glycine is important ratio in certain polypeptide turns too. And turn is another structure, the feature of the polypeptide that we're going to discuss much more in the later of this lecture. And the last one among this special amino acid is proline, Pro or P, which is a cyclic amino acid, and this is the only cyclic amino acid among 20 standard amino acids, where the side chain forms a covalent bond with the nitrogen atom in the amino group, and its cyclic properties make the proline rigid and difficult to fold into polypeptide chains. So it tends to create a fixed kinks, so-called an alpha helix breaker, that we're going to talk about it later on when we discuss about the secondary structure of a polypeptide. And proline often plays a role in certain polypeptide turns too. Again, we're going to discuss about all these polypeptide structures later on. Like we discussed earlier this lecture, all amino acids have handedness, meaning that they can have the mirror images which are not superimposable, and that property is called chirality or handedness. And chiral compounds are optically active, interestingly, which means that when there's a light source that goes through the fixed polarizer, so the polarized light will go through that particular compound within the polarimeter tube. In that case, these compounds can rotate the plane of a polarized light. So when you look at it with the analyzer, you can actually analyze that how much of the angles that this polarized light was tilted or turned. And that happens for all the chiral compounds. So that applies to all the amino acids except for the glycine, which does not have chirality. Enantiomers arise from compounds with chirality. So they are the stereoisomers that are not superimposable, which are the complete mirror images of each other. And it's the same as one's left and right hands that are the same, but at the opposite phase. Enantiomers have identical chemical and physical properties, except the fact that they have ability to rotate plane polarized light in opposite directions, like we've seen in the previous slide, but by equal amounts. And a mixture of the equal parts of an optically active isomer and its enantiomers is termed racemic. And this racemic, since it has the equal parts 50 and 50 of the enantiomers, It has a net rotation of a plane polarized light of 0. And interestingly, there is only L enantiomers of amino acids are made in the cell. But you can actually get the racemic mix, meaning that both the D and L form from the organic synthesis of amino acids in the lab. But the fact that there's only L enantiomers amino acids observed in the cell leads to the evidence of a stereospecificity of enzymes, meaning that there's an enzyme that is particularly evolved in a way that interacts only the L enantiomers of amino acids. An optical isomer can be named by the spatial configuration of its atoms, and Fischer convention, or the DL system, for the D and L forms does this by relating the molecule to glyceraldehyde. Here's the glyceraldehyde, which is a chiral itself too, because it has all different groups that are attached to the chiral center, which is the carbon inside here. And its two isomers are labeled as L and D glyceraldehyde. And Fischer projections actually simplifies this drawing from the geometric formulas by using only the bars or the lines. And horizontal bars, meaning that these groups are above the plane of the paper that is protruded towards you, as opposed to the vertical lines are basically indicating the groups that are behind the plane of the paper. Certain chemical manipulations can be performed on glyceraldehyde, so changing some of the constituents here without affecting its configuration. And its historical use for this purpose has resulted in its use for nomenclature for indicating the configuration of those modified or manipulated molecules too. In this system, compounds are named by analogy to glyceraldehyde configuration here, as you can see, as L or D form, which generally produces unambiguous designations. And it is easy to see in the small biomolecules similar to glyceraldehyde, such as amino acids, has a similar composition here. A rule of thumb for determining the D or L isomeric form of an amino acid is the Korn rule. There are 4 groups around the chiral center of a carbon atom within the tetrahedral structure of amino acids. The carboxylate group, amino group, hydrogen atom, as well as the R group, which is a variant carbon side chain. And CO from the Korn, or the Korn, CO was from the carboxylate, R was from the R group, and N was from the amino group to form this Korn rule. So to apply this Korn rule, first place the hydrogen atom, which is the smallest atom, away from the viewer, from you. And if these groups are arranged clockwise, so the Korn, like that, then it is the D form. If that is counterclockwise, like Korn, like that, then it is the L form. The 20 amino acids that we discussed earlier in this lecture are standard amino acids used by cells in protein biosynthesis. And these are specified by the general genetic codes called codons that we're going to discuss later in this lecture. 20 amino acids can be biosynthesized from simpler molecules, and organisms differ in how many they can produce. And the amino acids they cannot produce are called as essential amino acids that must be obtained in their diets. An amino acid residue is one amino acid that is joined to another by a peptide bond among the polypeptides. And each different protein has a unique sequence of amino acid residues. This is its primary structure. And of 20 standard proteinogenic amino acids, 10 are called essential amino acids, because the human body cannot make them by their own from other compounds, so it must be obtained from the food. So these are the 10 essential amino acids. And a helpful mnemonic for remembering these essential amino acids is provided Tim Ho. Well, help yourself to use this. And histidine and arginine are generally only considered as essential amino acids in children, but in not adults, because the metabolic pathways that synthesize these amino acids are not fully developed in children. Genetic code, which has codons, is a set of rules by which information encoded in genetic materials, such as DNA or RNA sequences, is translated into proteins, the amino acid sequences, by living cells. And the genetic code defines a mapping between trinucleotide sequences called codons and amino acids. So basically connects to these two different informations. And every triplet of nucleotides in a nucleic acid sequence specifies a single amino acid. For example, ATG in nucleic acid sequence specifies a methionine. And most organisms use a nearly universal code, the genetic code, that is referred to as the standard genetic code. Even viruses, too, which are not cellular and do not synthesize proteins themselves, have a protein made using this standard code, too. There are also non-standard amino acids, and two of these can be encoded in the genetic code, but are rare in proteins. The examples are the selenocysteine and pyrrolysine. And here's the RNA codon table, which shows all the possible genetic codes. So the first base in the codon can be written right here on the left-hand side. The second base is on the top, and the third base in the codon can be seen on this right-hand side. So for example, if you're looking for CUG, then C on the first codon is right here, and the U on the second base on the codon is right here, and the G is right here on the bottom of the second row. So it'll cause the leucine here as an amino acid that is matching. And that works exactly the same way for the translation, as there's a codon on the messenger RNA, and there's an anticodon that is matching on the transfer RNA, which is attached to the corresponding amino acid too. Multiple amino acids can be linked by the formation of the peptide bond, because one amino acid molecule has both the amine and carboxylic acid groups that can react to form the amide bonds or the peptide bonds. And one amino acid molecule can react with another and become joined through the amide linkage. And that's what creates the protein or the polypeptide. And this condensation of a reaction yields the newly formed polypeptide or the peptide bond formation. And there's a molecule of water that is also generated. In cells, peptide bond formation does not occur directly or spontaneously. Instead, the amino acid is activated by the attachment to the transfer RNA molecule through an ester bond. This amino acid tRNA is produced in an ATP-dependent manner, which is carried out by an amino acid tRNA synthase. And amino acid tRNA is a substrate for the ribosome, which is basically a factory for the protein, which catalyzes the formation of the peptide bond. As a result of this mechanism, all proteins are synthesized starting at the N-terminus, which is amino terminus, and moving towards to their C-terminus, the carboxylate terminus. Proteins are workers in the cells. They are catalysts, and they also have structural roles such as scaffold. They are transporters for the compounds, ions, and molecules, and so on too. And also they are regulators for cell signaling, which is important for the information processing within the cell, between the cell, and also within a tissue too. And proteins are the most abundant among all the macromolecules, constituting more than 50% dry weight of the cell. And there are also exotic proteins such as green fluorescence protein, or the GFP, from the jellyfish, luciferase of a firefly, antifreeze of an arctic fish, and honeybee melatonin, and so on too. And there are more structural diversities in the proteins than the nucleic acids, because there are 20 building blocks, at least 20 of them, amino acids, compared to the nucleic acids have only 4 nucleotides as building blocks. So For 100 amino acid long proteins, there can be a total combination of 20 to the power of 100 variants. There are possibilities of different sequences there, which is the same as 10 to the 130th. But there's only 10 to the 79th particles in the universe. So there are a lot of different combinations and possibilities for the sequence variations. There is also an atypical peptide bond formation. In some cases, peptides are synthesized by specific enzymes rather than ribosome and going through the tRNA route. For example, the tripeptide glutathione is an essential part of the defense of cell against oxidative stress, but this peptide is synthesized in these two steps from 3 amino acids. First step is that the gamma-glutamyl-cysteine synthase condenses the cysteine and glutamine through a peptide bond. And then by this glutathione synthase, the glycine is attached to that molecule to form this tripeptide glutathione. And it can be transported to the other regions for acting, again, as a cell defense. And there are various side chain modifications in proteins, and these are some examples of such modifications. Amino acids other than the 20 standard ones contained in the protein are usually formed by post-translational modifications, which occurs after protein synthesis or translation. And these modifications are often essential for the function and regulation of that protein. For example, hydroxylation of proline is critical for maintaining connective tissues and responding to oxygen starvation. And such modifications can also determine the localization of the protein too. An example is that the addition of long hydrophobic groups of amino acid can cause a protein to bind to a phospholipid membranes because it gives the hydrophobicity of that amino acids. And lastly, there are also non-protein amino acids which have biologically important roles, not just in humans but also in animals and plants too. The examples include glycine, gamma-aminobutyric acid or GABA, or the glutamate, which are all neurotransmitters. And also, many non-protein amino acids are used to synthesize other molecules too. An example is tryptophan, that is a precursor of a neurotransmitter serotonin. And also, thyroid hormones are alpha amino acids.